Greetings, Brookings Bio students. This is Mrs. Rydell. We're going to continue talking a little bit more about transport in cells in living things. And we need a couple of vocab words. And I know these are review for you. You should have done this in middle school. But we're going to talk a little bit about molecules dissolved in liquids. So the substance that is dissolved is called the solute. And the substance that you are dissolving that solute in, the liquid, is called the solvent. So if I was to take a little beaker of water and add something to it, like sugar molecules, for example, sugar molecules would dissolve in the liquid, spread out in the water, and we would call that the solute, and the liquid that's dissolving those solute molecules is called the solvent. So if I want to talk about comparing high concentration or low concentration, we need kind of a definition of concentration. And the science definition is the mass of a solute in a given volume. If you have a certain volume of liquid, how many solute molecules are dissolved in that liquid? So if I was going to make Kool-Aid, for example, and I was to make two different containers of Kool-Aid, and I put the same amount of water in both containers, but I add more solute to the container. If those little black polka dots represent something dissolved in the water, for example sugar, if I asked you to taste those two containers of Kool-Aid, which one would you say would taste sweeter? And if you look, the one where the most black polka dots are going to have the most sugar molecules. The one on the right would taste sweeter if you tasted that container. And so if you think about that idea of more molecules, the more molecules you have in a given volume, we would say that concentration is greater. The concentration of solute molecules, of sugar molecules in the right-hand container is greater than those in the left-hand container. There's more sugar molecules in that same space. So let's make a connection to something that you know. If you're making Kool-Aid, what would be the solutes in that little situation? Think about solutes. Remember, those are the things that are going to dissolve. So the solutes in Kool-Aid would be the Kool-Aid powder and the sugar. The solvent, the liquid that you dissolve those solutes in, would be the water. And when you put those two together, the solutes plus solvent, you get what we call a solution. And if you think about cells, cells are basically solutions of molecules, mostly water in your cytoplasm, in your blood, in your bodily fluids, and lots of little molecules dissolved in that water, in that liquid. You have carbs, you have proteins, all those different kinds of molecules, ATP, ions, all those different kinds of substances we talked about. You are basically a solution. A cell is basically a solution of molecules. So what if there's a difference in concentration? We talked about that idea of if you have a difference of concentration across a membrane, that diffusion can happen. Molecules will go from the high concentration to the low concentration side, as long as the molecules can fit through the membrane. But what if the solute molecules can't move across the membrane, or they can't move very quickly? So in this little diagram, you have two sides of the membrane. Here's my semi-permeable membrane in the middle. These big blue circles stand for solute, something that's dissolved in the water. And we're going to put water molecules in here. Sometimes we don't do that. We just kind of leave blank spaces to show where the water is. But these little red polka dots stand for water molecules. And if you put a difference in concentration, different solute, on this side compared to this side, those blue dots are going to want to move across to make the concentration equal. They're going to want to move from the left-hand side to the right. But if they can't get through the membrane, the other way you could make the concentrations on both sides equal, to kind of dilute these down and spread them out more to be like this concentration, would be to add water to the left-hand side and dilute down those molecules until the concentration is equal everywhere. If the blue molecules can't go, water will move instead until the concentration is equal. Water molecules will move from the right-hand side over to the left to try and dilute down those little blue polka dots. So we're going to watch a little video about how this might work in a cell.
Here's another beaker of water. A membrane is added to this one. water can flow freely across a semi-permeable membrane. Recall also that the movement of water across a semi-permeable membrane depends upon the concentration of solutes on either side of the membrane. Therefore, whether water moves into or out of a cell will depend on the relative concentration of solute inside versus outside the cell. Suppose a cell is placed in a hypertonic solution. This is a solution in which the concentration of solutes is greater than the solute concentration in the cell. Water diffuses out across the cell membrane and the cell shrivels up. Now think about a cell placed in a hypotonic solution. The concentration of solutes in a hypotonic solution is lower than the concentration of solutes in the cell. Again, the flow of water acts to even out the solute concentration on either side of the cell membrane. Water diffuses into the cell, causing it to swell. More and more water flows in until eventually the cell bursts. Finally, consider a cell placed in an isotonic solution. This is a solution with the same concentration of solutes as the cytoplasm. Although water flows in and out of the cell, there is no net flow in either direction. The cell neither shrinks nor swells.
So let's look at this again. Remember, we're going to use little big circles here to represent something dissolved in the water, and we're going to use the smaller little dots to represent water molecules. Remember, solute would want to go from where you have a high solute concentration, high big dots over here compared to here. They would want to move to the left. But if they can't move, if they can't get through the cell membrane, water's going to want to do the same thing. Look at all the little black dots for the water molecules here. Lots of water, high concentration of water on this side compared to lower water on this side. Water molecules are going to want to go from where you have a high concentration of water to low concentration of water. If the big yellow dots can't move, the water molecules can, and they're going to go across the membrane from a high water concentration here to a low water concentration. They're going to move toward the solute, where there's high solute. And that's going to change the level of the water, but notice when it does, it equals out the concentration on both sides. So let's look at some situations in cells. You have several possibilities. One situation could be where a cell is sitting in a liquid that has a lower concentration of dissolved solutes outside the cell compared to inside. So these three little diagrams, here's these big circles are cells, and think of these as a little container holding a liquid, and inside the liquid there are molecules dissolved. They're solutes. And in this situation, we're going to use these little black polka dots to represent solute molecules, something that's dissolved. It could be sugar, it could be an ion of some kind, it could be an amino acid, it could be any kind of a molecule. And so we're going to use this. Here's my little container, here's my cell, and notice now the polka dots, there's lots and lots of molecules inside of cells. Think about all those molecules you learned about: carbs, DNA, proteins, amino acids, ATP, ions that are inside cells compared to the liquid outside. Very few solutes, things dissolved in the water. That's these little black polka dots. We would say this cell is in a hypotonic liquid. Lower concentration of solutes outside compared to inside. And in these diagrams, we're not really showing you actual water molecules. All these kind of white spaces are where the water is. Okay? And the little black dots stand for something dissolved in the water. Hypo means less or below. Like when you get a shot at the doctor, they give you a shot with a hypodermic needle. It goes down below under your skin when it gives you the medicine. The other situation a cell might be in is where the concentration of solute molecules, things dissolved in the liquid, is equal outside and inside, equally spread out. Remember we said equilibrium. If the concentration of solute outside and inside are equal, we say the cell is in an isotonic liquid. And the other situation a cell might find itself in is if the concentration of solute, remember these are little things dissolved in the water, the little black polka dots, is greater outside the cell compared to inside the cell. There's lots and lots of molecules dissolved in this water compared to what's inside the cell. We would say the cell is in a hypertonic liquid. Hyper means above, over, more than. When you talk about someone being hyperactive, they're acting above or over the top. And so those situations cells can find themselves in can result in very different things happening to the cell. So we're going to take a look at what could happen to an animal cell if we put it in different liquids. And we're going to do a lab later on in the chapter that has to do with looking at what happens to cells if you put them into different kinds of solutions. So remember, cells like to maintain stable internal conditions. We learned that in our first chapter. We've talked about it several times. That idea of homeostasis, staying home, keep it steady. And so if I take an animal cell, like a blood cell, and I drop it into a liquid, if that liquid has the same concentration of solute molecules outside compared to inside, isotonic conditions, the water is going to equal on both sides. Water entering the cell is going to equal the water leaving the cell, and the cell is going to stay the same, stay at homeostasis. And blood cells, body cells, animal cells, they like to be in homeostasis. They like to be in isotonic liquids. Blood is isotonic to the cells. Sometimes, though, the cells end up in a situation where inside and outside concentrations are not equal. And if the cell can't maintain that homeostasis, keep the concentration steady, damage can result and the cell can die. So let's look at what might happen to an animal cell if I put it in some different liquids. Hypotonic situation, remember these little black dots stand for something dissolved in the water. If you look at this situation, here's my cell, lots of molecules in the cell, fewer molecules in the liquid outside. We would say this cell is in a hypotonic situation. Concentration of solute outside the cell is less than inside. And in that situation, remember, if the solute can't move, water is going to move from where there's high water. Look at all these blank spaces out here. High water concentration compared to less water concentration in the cell. Water is going to move into the cell. And more water enters than leaves the cell is going to get bigger, bigger, bigger. It can possibly explode. We're going to add something to our little picture here. I'm going to ask you to do this. And I'm going to write the word solute. Because solute really determines where the water is going to move to. Remember, it's going to move from a high water concentration area to a lower water, but really it's moving from where you have low solute to where you have high solute. I'm going to ask you to add this to your slideshow. Solute sucks. And my pen's not working very good here. Solute sucks. If you could imagine all of those little solute molecules as little Pac-Men with mouths, and they all have straws in their mouth, like a drinking straw, and they're pulling water molecules toward them. Each of these little solute molecules has a little straw in their mouth. And what's going to happen in this situation is solute sucks. Look where all the solute molecules are. They're going to pull water toward them. Will there be some water leaving? Yes. But the majority of water in this situation is going to move toward where the solute is. It's going to move from a high water concentration outside to a lower water concentration inside. It's going to move toward the solute. Solute sucks, and it pulls water molecules toward it, and it's going to cause the cell to get bigger and bigger and bigger and possibly explode. If we have a hypertonic situation, greater solute concentration outside the cell compared to inside, lots of solute molecules, could be salt, could be sugar, any kind of a molecule out here. Then we have high water here. Look at all the empty blank water spaces here compared to little solute. And you have high water in the cell compared to low water outside. Water molecules, remember, solute sucks. I'm going to add it again. Solute. Sorry for my bad handwriting. Solute sucks. My pen is not working. Imagine each of these little solute molecules having a drinking straw and pulling the water out of the cell. Some water will be entering. Not all the water leaves the cell. Some will be going in. But a way greater majority of water molecules leaving my cell, going toward the solute. And that is going to cause the cell to shrink smaller, shrivel up. We're going to watch a little video here that looks at what could happen now with the actual cell. These are videos that were taken with a microscope, and they placed human blood cells on a microscope slide and put it under the camera, and then looked to see what happened to the cells when they put them in different liquids. So let's take a look at blood cells in a hypertonic. There's my little blood cells, and notice as the water leaves the cell, that kind of crinkles up, wrinkles up, gets all bumpy and lumpy. You can see the cells starting to change their shape. If you put a blood cell in hypertonic, it will shrink and shrivel. How about if I put my cell in a hypotonic situation? Fewer molecules outside than in. And remember, that means water is going to enter my cell. I'm going to take those little shriveled up cells and add distilled water to them, and now they start to poof. They get bigger, bigger, bigger. Notice how big and round they are. And they actually start to pop. These ghost cells that you're seeing are the remains, the leftover cell membranes that are left behind after the cell has popped. Let's watch that again. We're going to add hypotonic. We're going to add some distilled water to those lumpy, bumpy cells and see if we can get them to poof up. Notice they go from lumpy, bumpy back to bigger, 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 bigger. They get nice and poofy, and they start to pop. Animal cells, if you take an animal cell like a blood cell and put it into distilled water, that is a very hypotonic situation. Remember, distilled water doesn't have very much stuff dissolved in it compared to the cell. And cells in that situation are going to take in water. They're going to swell bigger and possibly burst. We call that cytolysis. Cyto means cell. We had that word before, like cytoskeleton. And we also had the lysis word before. Remember, we had hydrolysis back in our first chapter, breaking molecules apart using water. We had lysosomes, those little cell parts that break stuff apart. So cytolysis, cytolysis, the breaking apart of cells. And cells that are placed in a concentrated salt solution, a hypertonic situation, causes the cells to shrink and shrivel up. And we have a word for that also. It's called crenation. Crenation. The cell is going to shrink and shrivel and get all lumpy and bumpy. It works the same for a plant cell. If you take a plant cell and put it into distilled water, it's going to cause the cell to take in water, and it's going to poof up and get very big and round. And but it doesn't explode like an animal cell. So think about what you know about plant cell parts, and what do plants have outside them to keep them sturdy, hold their shape, give them support. And because they have that cell wall, the plant cell doesn't explode. It doesn't burst like an animal cell, but it can take on water and poof up very big and cause lots of pressure to build up inside the cell. If you put a plant cell in hypertonic, where water is leaving the cell actually doesn't change shape because the cell wall is here, kind of like a brick wall, holding that position. But inside, the cell membrane, the plasma membrane, can actually shrink and shrivel and pull away from the cell wall. And we call that plasmolysis. Remember, we said plasma membrane is the membrane on the outside, and the plasma membrane is actually shrinking and pulling away from the cell wall. Plants actually kind of like being in hypotonic. When you water a plant with tap water, for example, there's not very many molecules in that water compared to a plant cell. If you've ever gone away on vacation forgot to water your plants for a while, you come home and see they have this kind of wilty, bent over shape. They don't have a lot of water pressure inside to help them stand up. But if you add water to the plant, if it hasn't been too long and the plant has died, if you add water to the plant, water the plant, it's going to take in that water and send it to its cells. And remember, vacuoles store water. That was our cell part. We're going to fill up the vacuoles in each cell, and the cell's going to poof up bigger and push against the cell wall, and the plant kind of stands up, pumps up. That pressure of water pushing against the cell wall is called osmotic pressure. Pressure of osmosis, pushing against water molecules, pushing against the cell. And it causes the plant to stand up and be very perky and firm. And so plants actually kind of like being in hypotonic a little bit. They like to have that water pressure inside holding them up. Also called turgor pressure. If you look on your vocab list, it says osmotic or turgor pressure. So what does this have to do with you? If you think about sitting in a bathtub, bathtub water is pretty hypotonic compared to what's in your body cells. Very few molecules dissolved out here compared to what's inside your cells. And after you sit in the bathtub for a little while, causes your fingers and toes to kind of wrinkle up because water is entering your skin cells. You have collagen and keratin in your cells, lots of other kinds of molecules in your skin, and that kind of holds the position of your fingers, but the cells kind of poof up around those fibers, and you get those kind of like ridges and little poofy areas. If you've ever sat in the bathtub for a really long time, or after you take a shower for a long time, you feel how poofy your skin feels, very hydrated. It's because water is actually entering your skin cells. And same thing happens in the grocery store. Have you ever seen them spraying water on the veggies? Nobody wants to buy limp broccoli or saggy celery. Spraying water on those veggies is putting hypotonic liquid on them, and they absorb the water, they take it into their cells, and it plumps them up. Think about a living thing, an organism that spends time in water. A freshwater fish is basically spending its whole life in a hypotonic situation. Ions are constantly leaving their body because you have a greater concentration of ions in the cells compared to the water. And water is constantly entering the fish's body. And if it can't do something to maintain homeostasis, keep the water balance and the ion balance in its body, that fish could die. And so what do freshwater fish do? They urinate, they pee constantly to get rid of that water that's entering, and they actively pump ions in from the water into their body to replace the ones that are lost. If you're a saltwater fish hang out in salty water. So ions are constantly entering their body through their gills, and they're constantly losing water out through the surface, out through their gills, and so they have to do the opposite. You can't take a freshwater fish and put it in the ocean. It will die. You can't do that change in balance. Saltwater fish, they urinate very rarely. They conserve their water. They hold on to it. They don't pee. And they actively pump those ions that are constantly entering out through their gills to maintain their homeostasis. And if they can't do that, those fish will end up with the imbalance in their water and their ions, and they can die from that situation. I think that's all we need to know, and we're going to stop right here. Thanks for watching.